홍용길 선생님 모시겠습니다.
시간이 돼서 지금 또 연자도 나와 계시고 그래서 오후 세션을 시작하도록 하겠습니다. Good afternoon, ladies and gentlemen. We'll start the first afternoon session, and the guest speaker, Dr. Mami Yamasaki. Dr. Yamasaki graduated Kyoto Prefectural Medical College in 1978, and she finished pediatrics between 1978 and 1982, and she had then finished the postgraduate course of neurosurgery in Osaka University Medical School, and she had been abroad for further study in Department of Neurosciences, Case Western Reserve University of Cleveland, Ohio, between 1996 and 1997. So today she will give us a talk about molecular biology of congenital hydrocephalus and guideline for fetal hydrocephalus. Dr. Yamasaki, please.
Good afternoon. Thank you very much to give me such a great opportunity to give my talk today. So I will start my paper. So my paper, I organize a research committee of intractable hydrocephalus funded by Ministry of Health, Labor and Welfare of Japan from 1999 to 2004. And our project focused to fetal hydrocephalus and molecular mechanism of congenital hydrocephalus. The clinical and ethical problems of fetal hydrocephalus continue to be unsolved. The aim of this paper is to introduce our result. So today's talk is two themes. Slides, and the first is the molecular biology of congenital hydrocephalus. We founded the DNA bank of congenital hydrocephalus, and we accumulated 558 samples of DNA. And the recent advance of molecular biology, so many candidate genes about the related congenital hydrocephalic syndrome was discovered. So today I will focus to L1-CAM gene. So 1992, Cambridge and her colleagues detected the L1-CAM gene mutation in a family with X-linked hydrocephalus. X-linked hydrocephalus is first reported in 1949 by Vickers and Adams. The hydrocephalus retardation, spastic paraplegia, and adducted sum is specific findings of this disease. Since then, 178 mutations have been found in 206 unrelated families, not only in XLH, but also in Martha syndrome and X-linked spastic paraplegia. These syndromes are now called as L1 syndrome. So what is L1-CAM? So L1-CAM has multiple aspects of neural development, such as neurite outgrowth, axon fasciculation, myelination, and migration of neural precursors. L1 is expressed developing central and peripheral nerves. So L1 is a 220 kilodalton molecule, member of immunoglobulin superfamily of cell adhesion molecule. L1 has a 6 Ig-like domain, and followed by a fibronectin type 3 domain at the extracellular domain, and single pass transmembrane part and cytoplasmic domain. So extracellular domain, they have a hetero-binding site and also a homophilic binding site. cytoplasmic domain, so they have a many signal transduction system. So L1, this is a cartoon-like schema of L1. They have a cytoplasmic domain and transmembrane part and extracellular domain. So L1 and L1 is homophilic binding, and to adhesion, the neurons each other. Homophilic transbinding of L1 molecule is the most common mode of action, like cell adhesion and migration. L1 mediated cell adhesion and migration requires extracellular domain function. And L1 mediated axon growth. So this is a growth cone, this is axon growth. So from the data of basic research of L1, L1 mediated axon growth required only L1CD function. So L1 gene is mapped on the XQ28 chromosome in humans. It's composed of 28 exons. The open reading frame has about 4,000 BSP, a very big gene. So, and the method, genomic DNA was isolated from peripheral blood. Amplification of the 28 exon intron border of the L1 CAM gene was performed by PCR. PCR product was verified and direct sequence was performed. So, and the mutation of L1 is, every mutation is have each different site and different types. So I classify the L1 gene mutations as 3 types, based on how they would affect the expression of structure of the protein. The first class of mutation affect only the cytoplasmic domain. The second class consists of missense point mutation in the extracellular domain. The third class of mutation include nonsense or frame shift mutation that produce a premature stop codon, and they don't have a transmembrane domain. So how does L1 mutation disturb the L1 function? This is a normal L1. So class 1 mutation is only here, but the extracellular domain works well. So the class 1 mutation disturbed axon growth only. But class 2 mutation that was extracellular domain doesn't work. So, and class 3 mutation doesn't stay cell surface. So these 2 mutation doesn't have a cell adhesion and migration and axon growth. And so in a sample, we did the L1 CAM gene analysis, and under the criteria for L1, one is patients with hydrocephalus with X-linked inheritance, and second is sporadic cases with clinical and neuroradiological characteristics of XLH. And we found 31 types of L1 mutation in 33 families with XLH. In these, the 7 was classified as class 2 mutation. This is an extracellular domain point mutation. So, but this point mutation, all of these in extracellular domain are key residue for keep the structure of extracellular domain. And the 7th case was classified as a class 3 nonsense mutations. And 5 families were classified a frame shift mutation, and it was categorized in class 3. And class 3, this 7 and 5, product of L1 protein was premature stop codon, so, and very short L1 is produced. So they don't have, due to loss of a transmembrane domain, L1 failed to remain at the cell surface. Splice site mutation was found in 12 cases. And RT-PCR was revealed that this mutation is this one, the splice site was mutated. So this part of was not spliced out. So abnormal long L1 was produced this one. So also this splice site mutation is also due to loss of the transmembrane domain. So L1 failed to remain at the cell surface. The 33 families with X-linked hydrocephalus had class 3 mutation and key regio mutation of class 2 and splice site mutations. This is all, the phenotype is a severe type of L1 syndrome. So L1 and hydrocephalus. So how this function of L1 make hydrocephalus? What function of L1 is responsible for hydrocephalus? So this type of mutation, class 2 mutation, not have a normal extracellular domain and premature stop codon, a short L1. Also, this type of L1 lose L1 extracellular domain function, so lose the L1 mediated cell adhesion and migration. So to clarify the mechanism of developed hydrocephalus, we performed clinical evaluation for severe type of L1 syndrome. These types of a very severe ventricular dilatation. So the most cases were diagnosed by fetal sonography or MRI. So in the fetus, lateral ventricular volume was increased at around 20 weeks, so very rapidly increasing. And they have another characteristic finding after VP shunt. They showed the ventricular wall showed the ripple-like, so very unusual phenomenon. So, and to look at precisely, they have enlarged massa intermediate and also enlarged quadrigeminal plate. So with the NIH images program, I performed the area measurement, so of a quadrigeminal plate. So the L1 severe type of L1 syndrome is significantly bigger than normal. So and also they have a hypoplasia over vermis. Some over total vermis hypoplasia. Some over anterior vermis only hypoplasia. So and also statistic very small than normal. Hypoplasia of the vermis and enlarged quadrigeminal plate, enlarged massa intermediate. These are associated with midline maldevelopment of CNS, perhaps involving abnormal neural migration. So this family was have a splice site abnormal. So the second boy was diagnosed as L1 syndrome. So he was aborted, and so under the approval of use of fetal neural stem cells obtained by my ESCAL committee of my hospital. So we made isolated from post-mortem autopsy brain tissue. So culture started after 20 hours post-mortem time. So we succeeded to isolate human neural stem cells with L1 mutations. So that result, a cellular doubling time of neural stem cells of XLH is shorter than normal controls. Compared to the normal is 82.5 hours. The XLH showed 64.2 hours. So proliferative activity is XLH is more than normal. So and another data is real-time quantitative PCR examination. So transcripts of 4 well-known neurodevelopment marker genes examined by real-time quantitative RT-PCR. So the red is XLH, blue is normal. So the neural stem cell precursor cells is the RNA, the volume is increasing. So, but the L1 XLH is not increasing. So, and tubulin beta 3 is very decreased, and also GFAP and PAX3, PAX6. And so look at this double coaching. Double coaching is a migrate neurons marker, so it is almost lax to increase. So, and another data is differential assay using the confocal images. The neurosphere to histocyte chemistry was done. So red is tubulin beta 3 and GFAP is red. So green is tubulin. So the compared to normal, so XLH is axon are shorter and the shape of glia is abnormal. Also the differential ability is also defective. So conclusion, neural stem cells with L1 mutations, proliferative activity increase, differentiation ability defect, neural cell migration is lack. So we estimated the molecular basis of hydrocephalus of L1 syndrome. So the neural stem cells migrate to the from subependymal to the surface of cortex. To the loss of L1 mediated migration and the proliferation of neural stem cells increase here, and so enlarged massa intermedia and enlarged quadrigeminal plate. So they bring a narrow of the third ventricle or a narrow of aqueduct. So that will be produce a narrow CSF space. So, and the second molecular mechanism of hydrocephalus, we estimated loss of L1 mediated adhesion. In the white matter, the axon and adhesion was decreased and L1 lax. So compliance is increased. Increase the vulnerability of the ventricular system. Irregular shape, the lip rod shape of after shunt is due to this phenomenon. So beside of class 1 mutation, so we found this boy L1 mutation in cytoplasmic domain. They don't have a hydrocephalus. So they have only bilateral adducted thumb and mental retardation. So from this case, so adducted thumb was caused by a loss of innervation of extensor polystic muscle, and also hypoplasia of corpus callosum. This phenomenon is observed in all cases of the L1 syndrome, not with the hydrocephalus also. So these are also observed in L1CD mutation Martha syndrome. So adducted thumb and hypoplasia of corpus callosum due to a disturbance of axon growth. So, and L1-mediated axon growth require L1-CD function. So this is normal. The growth cone is axon growth. So, and class 1 mutation and also class 3 mutation disturbed this function. So conclusion, X-linked hydrocephalus is a severe type A1 syndrome. X-linked shows severe ventricular dilatation, a ripple ventricular wall after shunting, hypoplasia of the cerebral vermis, anterior total, an enlarged quadrigemini plate and a large massa intermediate. This study further confirmed that the disruption of L1-ED function regions, L1-mediated cell adhesion and migration is responsible for progressive ventricular dilatation in humans. L1-CD function is important for L1-mediated axon growth. So prenatal diagnosis of L1-COMG mutation is X-linked hydrocephalus. So it is very clinical. And the simple hydrocephalus does not always show poor outcome. That this girl is also a simple hydrocephalus diagnosed by in utero. So she is now studying, working as a nurse. So she is entirely normal. So, but X-linked hydrocephalus is very poor outcome. And X-linked inheritance, so 50% of male fetus are healthy, and 50% of male fetus is XLH, and 50% of female fetus are healthy carrier, and 50% female fetus not carry mutations. So from Tokyo University, the first we are ordered to prenatal diagnosis. So the first boy was born with severe hydrocephalus with family history. So, and detected in the family L1 mutation. Mother was pregnant and wanted gene analysis. CVB was detected done at 10 weeks gestation, male fetus in chromosome analysis. No L1 mutation, so continued pregnancy, healthy male baby was born. So we performed in 5 families of prenatal L1 analysis. So the diagnosis was made with perfect accuracy. No maternal fetal complication is chronic. A chorionic virus biopsy was occurred. Prenatal L1 gene analysis are helpful for the families with X-linked hydrocephalus. So, and another topics to my report. So we did nationwide surveillance for congenital and fetal hydrocephalus. That result also showed the basic disease of congenital hydrocephalus was very the basic disease. The 1/3 is 36% is hydrocephalus with myelomeningocele. A simple hydrocephalus is 40%. The outcome of fetal hydrocephalus had a very wide variety, and 90% is normal, and subnormal is 22%. So, but some of them, 6% is die, and 28% is have a very severe retardation. So we analyze data about prognosis of every individual basic disease. So simple hydrocephalus. Simple hydrocephalus includes the X-linked hydrocephalus or some another non-hereditary hydrocephalus. So that have a very bad prognosis is so, but some is very good prognosis. It's very difficult to estimate. So gene analysis or a chromosome analysis is very helpful to this simple hydrocephalus. About the look at myelomeningocele. So some patient was died with Chiari malformation, but the relatively good outcome in myelomeningocele. Dandy-Walker syndrome is 6%, but so they have also variety of outcome. Encephalocele is separated two types. So very good outcome or very bad outcome. Holoprosencephaly is very bad. All of the type of holoprosencephaly shows a very bad outcome. Arachnosis is only 2%, but so they have a good outcome. So we need to attention. So completely different outcome between interhemispheric arachnosis and holoprosencephaly. This is an interhemispheric arachnosis, and this is a semi-lobar holoprosencephaly. The outcome is completely different. So they are some secondary fetal hydrocephalus injury arising in utero, brain tumor, or intracranial hemorrhage in utero, or hydroanencephaly caused by infection or infection. So in Japan, it is increasing in the ratio of prenatal diagnosis in congenital hydrocephalus to three times. So it is because of fetal ultrasonograph have become routine work, and the improvement of the diagnostic equipment have done. So when fetus was diagnosed with hydrocephalus, parents, especially mothers, are shocked with sadness. Most obstetricians are also confused because they do not have enough information about outcome of fetus. Ambiguous or uncertain information make parents over-anxious. Accurate, objective diagnosis and information of the fetus and counseling and supporting system for the parents are required. Afterward, they come to face to reality and go on to delivery with courage. Not only ethical problem, clinical problem also have been left unsolved about fetal hydrocephalus. When is appropriate time to deliver? What is the method of delivery? Or when is the best time to treat hydrocephalus with meningocele? Should the weight of the body weight over 2,000 grams for the shunt surgery? So we in 2005 we published a guideline for the diagnosis and management of fetal hydrocephalus. This is for obstetrician, neurosurgeon, pediatric neonatologists and radiologists who are in the forefront of diagnosis and management of fetal hydrocephalus. And this is also to support the mothers. So thank you for my cooperated and banks. So, and the guideline was helped many doctors and Shirane and Liskopu is obstetrician and Noto Morota. So, and I will introduce my colleague, Department of Neurosurgery of Osaka National Hospital, and Clinical Institute, Research Institute of the members. So, and L1 research project done by Miss Shofuda and Hirai, and the neural stem cells work was done by Mori, and Dr. Kanemura is my best colleague. So he is a very good work and very organized over research. And so I'm afraid my English is poor, so, and if you have a question, please mail to me, Yamasaki at ONH.GOJP. Thank you very much for your attention.
Thank you, Dr. Yamasaki, for your excellent lecture on the congenital hydrocephalus. Because of time limit, I think we should go on for the next speaker.
Okay. I'm Dr. Hong from Catholic University. I'm honored to introduce Dr. Hee-Sup Shin. Dr. Shin has a more unique career as compared to the general medical doctors in Korea. Dr. Shin initially graduated from the College of Liberal Arts and Science, Seoul National University in 1970. And then he entered the medical school of Seoul National University and graduated in 1974. And then he started basic science research in Seoul Graduate School of Seoul National University, and moved on United States. And from 1978 to 1980, he started his postdoc fellowship in Sloan Kettering Institute for Cancer Research. And then he started his professional career as an assistant professor in Department of Biology, MIT, from 1955 to 1991. And then he returned back to Korea 1991 and as a professor of Department of Life Science Postdoc, Pohang University of Science and Technology. He served there 10 years, and he moved to KAIST, Korean Institute of Science and Technology. And since 2005, he became a director, a Center for Neuroscience. And thanks to the great scientific achievements, he's got many honors and awards. And at last year, he became a national honor scientist, which is first in Korea. And he's also doing many community service. He's now editor-in-chief the journal Molecules and Cells. And he was a member of the Presidential Advisory Council on Science and Technology Korea. And also now he's the president of Korean Society for Molecular and Cellular Biology. He's going to talk about understanding brain functions through mouse genetics. Dr. Shin, please.
Well, thank you for the introduction. Since we don't have any time, let's go to the first slide. So I'm a mouse genetics by training, and using this mouse genetics to study the brain functions. Next slide. We all know that the brain or individual is the product of genetic blueprint plus environment and experiences. And that's true for the brain also. Next slide. So hopefully we can understand human brains by studying animal models. There is a rationale for that. That is, the majority of human genes are also present in animals and function in similar ways. But how similar are they? That's the question. Next slide. I don't know if you do you agree? Next slide. Next. Okay, I guess pretty similar. Next. Okay, next. Sometimes the animal can be frustrated. Next. Or depressed. Next slide. Animals love to play like human being. Next slide. So the animal toy, that's one of the business. Next. You can click on this. So they love to play. Okay. Next slide. Next. So to study this kind of problem, mouse is a very useful model, especially for genetic analysis, because they are very small. Among the mammals, that's the smallest, among the smallest ones. You need a lot of samples to do genetics, like Mendel used the peas. And mouse, the little size is about 6 to 12, so it's very convenient. And generation time is very short. For 1 year, you can have about 4 generations breeding. So you can have result fairly quickly compared to other mammals. Okay, next slide. Gene knockout, that's the standard technology. That's helped a lot. I will just briefly, I'm sure you are familiar with this. Embryonic stem cell is the major tool here. In culture, in embryonic stem cells, some gene in the chromosome is replaced with some mutated gene. So the gene is mutated in embryonic stem cells. Then this embryonic stem cells is introduced into a blastocyst. It's all on the microscope. Into the blastocyst. Blastocyst consists of inner cell mass and the extracellular part, two populations. So this introduced cells will mix with this inner cell mass. They will become a mouse. So this embryo is transferred to the uterus, foster mother uterus. So they will grow there to become a chimera. So two cell populations are mixed. So coat color, in case of coat color, you can see that. Also, if this is a male, the sperm will consist of two populations. So if those males are crossed to some wild type, then some of the progeny will be derived from the sperm that was derived from the embryonic stem cells. So the mutation in embryonic stem cells will be transferred into chimera, then into the germline. So once you have that, you can breed them to make homozygote, then you have a mutant mouse for the gene you want it to mutate. Okay, next slide. So it's a convenient tool because you already know the molecular defect. You know which gene is defective because that's what you mutated. Then you see the phenotype in the mouse. So you see molecular defect and phenotype, which is causally related. So in the same body, so you essentially want to find out the process from the genes to behavior. Then if there is any behavioral change, that must be the result of some change in the brain which was resulted from the mutation. So genes, brain, and behavior, that's essentially what we like to study. So we can use this kind of approach to understand the neural mechanism for lots of brain functions. And the side product is often the target validation for drug development. So I will explain some of these things next. So genes to behavior. So you have a gene defect, then protein will be defective. Next slide. So the protein is active in some neuronal cells, then the neuron will be defective, then the neural circuit will be defective, then the result is the abnormal behavior. Next slide. We have been using this technique to study some genes in the neuronal cells that are involved in the regulation of intracellular calcium. Intracellular calcium is an important regulator of the neuronal cell excitability. Basal level is almost none, very little. Then when the neuron is activated, about 10,000-fold increase of calcium will be resulted. Then those increased calcium will activate lots of cellular processes in enzymes or receptors and so forth. And then the phenomenon will be driving all different functions of the neurons. For example, neurotransmitter release, or excitability, or neuronal metabolism, or gene transcription, in case of muscle contraction or proliferation. Lots of things can happen when calcium is elevated. So there are the genes and proteins involved in the regulation of this calcium. The calcium comes in from the outside through the channels, some voltage-gated channels, or it comes from inside calcium reservoir. That's the downstream of neurotransmitter receptors, metabotropic glutamate receptor, for example, and then PLC beta will be activated, then the IP3 produced, then calcium will be released from the internal store. So this calcium is very critical. But if this calcium stays too long, then it will be dangerous to the cell death. So that calcium has to be removed as quickly as it goes up. Calcium ATPase or sodium calcium exchanger. So bring the cells down to the basal level, then ready for the next stimulation. So we have been studying a group of voltage-gated calcium channels and some PLC beta enzymes and sodium calcium exchangers. Next. These are a list of the genes we have been working on. PLC beta 1, PLC beta 4 among the PLC enzymes. Sodium calcium exchange, we have all three of them. for example, there's the phenotypes listed, for example, epilepsy or enhanced pain, lack of innate fear, fetal rhythm, recently schizophrenia. The PRC beta 4 also has several phenotypes. Sodium calcium exchanger, NCX2, interestingly, they show enhanced learning and memory. So I will explain some of these things. Next slide. Calcium channels, also our favorite topics. There are many different calcium channels, and then each of them, when mutated, shows unique phenotypes. Or for example, alpha-1A ataxia, or severe absence seizures. Alpha-1B shows a pain response or increased aggression, or learning and memory is impaired. Empathy, alpha-1C, we recently found is involved in the empathy phenomenon. Here, alpha-1G is a T-type, shows several interesting things, and I just highlighted the 3 things that's related to the pain behavior. Okay, next slide. For example, aggression. How is the aggression controlled? Aggression is very necessary for survival in nature, but if it's not controlled, then it will cause lots of problem in society. Next slide. Click here. So when the male is isolated and another male is added into the cage, then something will happen. If a female is added, that's okay. It's peaceful. But where intruder is a male, then there is a big problem, because the resident male will attack, and they really fight a lot. And this aggression can be quantitative. For example, how early it attacks, how late it attacks. So one of the mutations we have shows increased aggressions, and we figure out why by analyzing brain functions in different brain regions. Next slide. Or mouse can solve some problems, maze problems. Next slide. For example, in the water maze, there is the tank here in the white paint. Inside is not visible, and the mouse is put it here, then it will swim around. And on the wall, there's some big visible markers, and the videotaping is done. Next slide. Then, next, underneath of the water surface, there is a hidden platform. So once the mouse realizes this platform is there, then next time when the mouse is added here, then it will go to find out where it is, and then it sits on it, because it doesn't really like to swim. And by training, the time it takes the mouse to reach the platform becomes shorter and shorter. Next slide. Before training, for example, it will go around, but after training, it will go directly. Next slide. So this is a latency to finding the platform, and training days. This is a wild type. First day, about 40 seconds something. It takes about that long for the mouse to find the place. And then next day already it's much shorter and becomes shorter and shorter. And the smart mouse we generated by mutation of one of the gene. In the first day is about the same, but the next day it's much faster in finding the hidden platform. So it already learned very quickly, and it learned how to navigate. So it's a spatial learning. So we figure out why the hippocampal function is enhanced in this case. But the important thing is, whether it's smarter or not, if they keep working, it will reach the same place. So we all know, right? Intelligence doesn't really guarantee. Working hard will be important. Next slide. You can also measure how long the memory lasts. After training, about a month later, platform was here, and it's removed now. It was there when training. Then a mouse with a good memory will go to that platform area and spin around to find out. Whereas a mouse with a bad memory, again, wandering all over the place. So this is the kind of assay you can use, and this is how we define a mouse has a better memory or defective memory. Next. You can click on here. So it's a videotaping of one of the performers. This is a mouse with a bad memory. Platform might be somewhere around here, and it cannot really find it. It takes so long. Right. On the other hand, if the mouse is a smart mouse, then they can find the platform very quickly. Next slide. Next, okay. So you can press here. So how about fear, emotional things? Fear is an important survival strategy, as you can see. There's a tunnel, there's a mouse, and there's a rat. Rat is bigger. This is a baby rat. It's not very big. But this mouse already feels some fear because a small animal is afraid of a bigger animal because usually they are predator. But curiosity is so strong, it keeps trying to figure out what it is, but fear rise again. But curiosity keeps coming back. So this is a typical response to some novel thing. You have a curiosity, at the same time you have fear. So finally I think it's going away. On the other hand, yeah, this one still tries. Eh? So when the mouse has anxiety and fear, they are usually drop some feces. So one can actually count the number of these. That's proportional to the anxiety and fear. Now, we have a mutant that seems to be fearless. This one. There's no fear. And this mouse can smell by other tests. There's a glass barrier here. So maybe we can remove this glass barrier and repeat the experiments. In fact, when we do that, there's no glass barrier. The rat is here, and the same mutant mouse. So it will go no fear at all. So this is by a mutation of one gene. So you can imagine what will happen if this rat is replaced by a cat. So appropriate amount of fear, that's a very essential part of survival. We all know that. Courageous and fearless, they are different things, right? Courageous people, they have fear, but they overcome. Fearless people, they just don't know. They just don't have the fear. And most of them, and most of those mice that were fearless, they all gone during the evolution in the nature. And in the same thing in human society, most of them are ended up in jail, as we know. Okay, next slide. One of the mouse shows some typical phenotypes of schizophrenia. It's a barbering behavior, social withdrawal, in other words. When the mouse are kept together, they lick each other and clip the whiskers. So they call it barbering. But when the mutant, they don't do that. So you see the large whiskers. If you mix one mutant and wild type, and all the whiskers in the mutant will be removed by the wild type, but the wild type will have all the whiskers left. So that's one of the social withdrawal. Next slide. So that's Pierce-Barrow knockout mice. They show many endophenotypes of schizophrenia. So it shows hyperactivity in a novel environment. And it shows social withdrawal. One of them is lack of barbering. And nesting behavior is defective and socially recessive. And also shows cognitive impairment like working memory. And it has abnormal sensory motor gating. PPI is impaired. And fetal rhythm is also impaired. And these phenotypes can be rescued by haloperidol, schizophrenia drug. Okay. So this kind of studies lead to some validation of drug development targets. You can stick here. So if some mouse shows some certain phenotypes, and you can use that as a model. If the when one gene is mutated, and you see some phenotypes, then those correlation can be used as a basis for developing drugs. For example, next slide. Pain. I'll take some example from pain signals. T-type calcium channels in pain regulation. Next slide. We have T2. There are several T-type channel genes, and two of them we have a mutation, alpha-1G and alpha-1H, and we analyzed their sensitivity in various pain behavioral assays. There are several pain behavioral assays, including normal nociceptive pain and inflammation and neuropathic pain. Next slide. Alpha-1G first. Next. This is electrophysiology, just to show that this is a channel. Alpha-1G is a low threshold channel. Electrophysiology shows that those channel function is gone. The other channel is okay. Next slide. I'm using all those molecular biology and electrophysiology. You confirm this mutation. Then behavior test, in the pain behavior test, von Frey test is a mechanical pain, no difference between wild type and mutant. Tail withdrawal from the hot plate or hot water, if there's a thermal pain, there's no difference between wild type and mutant. So acute pain, there's no difference. Next slide. On the other hand, visceral pain, which can be induced by peritoneal injection of acetic acid or magnesium sulfate, then the mouse will show abdominal stretching, some certain behavior. This is an assay some drug company use for developing pain drug. You can see that time and number of riding, a pain response. Wild type shows certain the pain response, and mutant shows enhanced pain response, hyperalgesia. Next slide. So we test whether this is due to some brain function, some specific region of the brain, and turns out it is due to the thalamic function. I will just go through the next slide. So in cartoon-wise, regular sensory information goes through the thalamus to the cortex. And when something persistent pain comes along, those information will be blocked in the thalamus. That's the function of alpha-1G, the T-type channels. So in the mutant, alpha-1G is gone, then the pain will keep going to the cortex. That's why it has hyperalgesia. Next slide. So in the thalamus, there is a mechanism by alpha-1G T channels to inhibit the persistent pain signals. But in the literature, T-type, there is a sign, evidence that T-type channel might be involved in the pain transmission. So the next slide. So we examined the alpha-1H, the other T-type channels. Next. And turns out this mouse, alpha-1H knockout mice, shows reduced pain for all mechanical, thermal, or chemical stimuli. They are just assays. So it's reduced for all the pain. Next slide. And turns out, unlike alpha-1G, this alpha-1H also shows reduced pain for the visceral pain, chemically induced visceral pain. So opposite to alpha-1G. Next. And so it looks like in the peripheral, DRG level, alpha-1H is involved in the pain signal enhancement. So when this is mutated, all the pain will be decreased. Whereas the alpha-1G in the thalamus is involved in signal blocking. So when this is mutated, pain will go, more information will go to the cortex. So it has hyperalgesia. Next slide. And then finally, we examined the neuropathic pain and alpha-1G and alpha-1H and their role in the neuropathic pain. Next slide. So neuropathic pain is due to a dysfunction or damage to a nerve. Three phenotypes: spontaneous pain, or allodynia, or hyperalgesia. Those are the phenotypes. Next slide. One of the models is that when the peripheral nerve is injured or the certain different part of the nerve is injured, then ectopic charge will generate some central sensitization in the spinal cord. That's one of the proposed mechanisms. Next slide. So there is a experimental model, Chung's model, that ligate the spinal nerve like this, then as time goes by, neuropathic pain will be induced. We used this approach. Next slide. Turns out alpha-1G and alpha-1H, they show different response. In alpha-1G knockout, compared to the wild type, mutant shows decreased neuropathic pain, whereas alpha-1H, there is no difference between mutant and wild type. Next slide. Also, mechanical allodynia also shows decreased response in the alpha-1G knockout compared to the mutant, whereas alpha-1H, there is no difference. Next slide. Thermal hypoalgesia also same. The alpha-1G shows less response compared to the wild type, so with the latency, so it takes longer. So alpha-1H has no effect. So in other words, alpha-1G seems to be involved in the generation of neuropathic pain. So in the mutant, which is lacking alpha-1G, shows less neuropathic pain compared to the wild type. Next slide. So this pain behavior and the T-type channel can be summarized like this. In the periphery, the information will go through DRG to the spinal cord, then from the spinal cord to the thalamic cortical relay neuron, then goes to the cortex. And in the peripheral DRG, alpha-1H is necessary for the pain transmission. It's a pro-nociceptive. In the spinal cord, neuropathic pain generation is dependent on alpha-1G. When the alpha-1G is gone, then it is impaired. And in the thalamus, alpha-1G is involved in the suppressing persistent pain from going to the cortex. Next slide. So this means when you have a T-type channel blockers, you may be able to control pain. For example, if you have a blocker that works at the spinal cord level, then it will reduce neuropathic pain. In the periphery, all other pains entry will be reduced if you block in the periphery. Okay, next slide. So in fact, we have a project going on at KIST trying to develop analgesia based on the T-type calcium channel blocker. Okay, and I won't go into that. I will stop here and thank you. And again, I'd like to congratulate the 50th anniversary of the Department of Neurosurgery here. Thank you.
Thank you for your excellent and very interesting lectures, and congratulate again for your outstanding achievement. We'll close this session.
감사장 전달이 있겠습니다. 먼저 Dr. Yamasaki. Dr. Mami Yamasaki, on behalf of the Department of Neurosurgery, Seoul National University College of Medicine, I take a great pleasure in presenting you this letter of appreciation in acknowledgement of your lectures, Molecular Biology of Congenital Hydrocephalus and Guideline for Fetal Hydrocephalus, given to our faculty and colleagues on September 1st, 2007 at this college. Dong-Gyu Kim, Professor and Chairman, Department of Neurosurgery, Seoul National University College of Medicine. Dr. Hee-Sup Shin, on behalf of the Department of Neurosurgery, Seoul National University College of Medicine, I take a great pleasure in presenting you this letter of appreciation in acknowledgement of your lectures, Understanding Brain Function Through Mouse Genetics, given to our faculty and colleagues on September 1st, 2007 at this college. Dong-Gyu Kim, Professor and Chairman, Department of Neurosurgery, Seoul National University College of Medicine.